Hello friends, welcome to Medical U. In this video, we will read the chapter 20 of 11th NCERT, that is Locomotion and Movement. So without further delay, let us discuss this topic. Movement is one of the significant features of living beings. Animals and plants exhibit a wide range of movements. Streaming of protoplasm in the unicellular organism like amoeba is a simple form of movement. Movement of cilia, flagella, and tentacles are shown by many organisms. Human being can move limbs, jaws, eyelids, tongue, etc. Some of the movement result in change of place or location. Such voluntary movement are called locomotion. Walking, running, climbing, flying, swimming are all some form of locomotive movement. Locomotive structures need not be different from those affecting other types of movements. For example, in paramecium, cilia helps in the movement of food through cytopharynx and in locomotion as well. Hydra can use its tentacle for capturing its prey and also use them for locomotion. We use limb for changes in body postures and locomotion as well. The above observation suggests that movement and locomotion cannot be studied separately. The two may be linked by stating that all locomotions are movement, but all movements are not locomotions. Locomotion means change in position, while in movement there is no change in position. While sitting, we move hands. So this is called a movement, not locomotion. Here you have to remember these two terms. Let us see the next. Methods of locomotion performed by animals vary with their habitats and the demand of the situation. However, locomotion is generally for search of food, shelter, mate, suitable breeding grounds, favorable climatic condition, or to escape from enemies, predators. Types of movement. Cells of the human body exhibit three main types of movement, namely amoeboid, ciliary, and muscular. Some specialized cells in our body like macrophages and leukocytes in the blood exhibit amoeboid movement. It is affected by pseudopodia formed by the streaming of protoplasm, as in amoeba. Cytoskeletal elements like microfilaments are also involved in amoeboid movement. Ciliary movement occur in most of our internal tubular organs, which are lined by ciliary epithelium. The coordinated movement of cilia in the trachea help us in removing dust particles and some of the foreign substances inhaled along with the atmospheric air. Passage of ova through the female reproductive tract is also facilitated by the ciliary movement. Movement of our limbs, jaw, tongue, etc. require muscular movement. The contractile property of muscles are effectively used for locomotion and other movements. By human beings and majority of multicellular organisms, locomotion requires a perfect coordinate activity of muscular, skeletal, and neural system. In this chapter, you will learn about the type of muscle, their structure, mechanism of their contraction, and important aspect of the skeletal system. Muscles. You have studied in chapter 8 that the cilia and flagella are the outgrowth of the cell membrane. Flagellar movement help in the swimming of spermatozoa, maintenance of water current in the canal system of sponges, and in locomotion of protozoans like euglena. Muscle is a specialized tissue of mesodermal origin. About 40 to 50% of the body weight of human adult is contributed by muscles. They have special properties like excitability, contractility, extensibility, and elasticity. Muscles have been classified using different criteria, namely location, appearance, and nature of regulation of their activities. Based on their location, three types of muscles are identified. First is skeletal, second visceral, and third is cardiac. Skeletal muscles are closely associated with the skeletal component of the body. They have a stripped appearance under the microscope and hence are called stripped muscles. As their activities are under the voluntary control of the nervous system, they are known as voluntary muscles too. They are primarily involved in locomotory actions and change of body postures. Visceral muscles are located in the inner wall of hollow visceral organs of the body like the alimentary canal, reproductive tract, etc. They do not exhibit any striation, and are smooth in appearance. Hence, they are called smooth muscles, non-striated muscles. Their activity are not under the voluntary control of the nervous system and are therefore known as involuntary muscles. They assist, for example, in the transportation of food through the digestive tract and gametes through the genital tract. As the name suggests, cardiac muscles are the muscles of earth. Many cardiac muscles help assemble in a branching pattern to form a cardiac muscle. Based on appearance, cardiac muscles are striated. They are involuntary in nature as the nervous system does not control their activities directly. Let us examine a skeletal muscle in detail to understand the structure and the mechanism of contraction. Each organized skeletal muscle in our body is made up of number of muscle bundles or fascicles held together by a common collagenous connective tissue layer called a fascia. Each muscle bundle contains a number of muscle fibers. Each muscle fiber is lined by the plasma membrane called sarcolemma enclosing the sarcoplasm. Muscle fibers is a syncytium as the sarcoplasm contains many nuclei. Syncytium means many nuclei. The endoplasmic reticulum, that is sarcoplasmic reticulum of the muscle fiber, is the storehouse of calcium ions. A characteristic feature of the muscle fiber is the presence of a large number of parallelly aligned filaments in the sarcoplasm called myofilament or myofibrils. Each myofibril has alternate dark and light bands on it. A detailed study of the myofibril has established that the striated appearance is due to the distribution pattern of two important proteins, actin and myosin. The light band contains actin and is called I band or isotropic band, whereas the dark band called A or anisotropic band contains myosin. Both the proteins are arranged as a rod-like structure parallel to each other and also to the longitudinal axis of the myofibrils. Actin filaments are thinner as compared to the myosin filaments, hence are commonly called thin and thick filament respectively. In the center of each I band is an elastic fiber called a Z-line which bisects it. The thin filaments are firmly attached to the Z-line. The thick filament is the A band are also held together in the middle of this band by a thin fibrous membrane called a M-line. The A and I bands are arranged alternately throughout the length of the myofibril. The portion of the myofibril between two successive Z lines is considered as functional unit of contraction and is called the sarcomere. Figure 20.2 represents a sarcomere which contains an I band, A band and a Z line. This complete structure is called a sarcomere. In a resting state, the edges of the thin filament of their either side of the thick filament partially overlap. The free ends of the thick filament leaving the central part of the thick filaments. This central part of thick filament not overlapped by thin filament is called the Z-zone. H-zone. The central part of thick filament, which is not overlapped by the thin filament, is called as H-zone. Here they have represented the H-zone, which is not overlapped by the thick filaments. Let us see the structure of contractile proteins. Each actin thin filament is made up of two F filamentous actin helically wound to each other. Each F actin is a polymer of monomeric G globular actin, two filament of another protein. Tropomyosin also runs close to the F actins throughout its length. A complex protein, troponin, is distributed at regular intervals of the tropomyosin. In the resting state, a subunit of troponin masks the active binding site for myosin on the actin filament. Here you have to remember that at resting stage, the subunit of troponin masks the active site of myosin. Each myosin, thick filament, myosin means thick filament, is also a polymerized protein. Many monomeric proteins called meromyosins Figure 20.3b. Myosin monomer or meromyosin. Here they have represented the structure of meromyosin. A cross-arm head, actin binding site, ATP binding site. Consists one thick filament. Each meromyosin has two important parts, a globular head with a short arm and a tail, the former being called the heavy meromyosin, that is HMM, and the latter the light meromyosin, LMM. The heavy meromyosin component, that is the head and a short arm, project outward at regular distance and angle from each other from the surface of a polymerized myosin filament and is known as cross-arm. The globular head is an active ATPase enzyme and has binding site for ATP and active site for actin. Here they have represented an actin, which is a thin filament which has troponin, tropomyosin and F-actin. And second is myosin, which is thick filament. Mechanism of muscle contraction. Mechanism of muscle contraction is best explained by the sliding filament theory, which states that contraction of muscle fiber takes place by the sliding of the thin filament over the thick filaments. Muscle contraction is initiated by a single set by the central nervous system CNS via motor neuron. A motor neuron, along with the muscle fibers connected to it, constitute a motor unit. The junction between a motor neuron and the sarcolemma of the muscle fiber is called the neuromuscular junction or motor end plate. A neural signal reaching this junction releases a neurotransmitter, acetylcholine, which generates an action potential in the sarcolemma. This spread through the muscle fibers and causes the release of calcium ions into the sarcoplasm. Increase in calcium level leads to the binding of calcium with the subunit of troponin on actin filament and thereby remove the masking of active site for myosin. Utilizing the energy for ATP hydrolysis, the myosin head now bind to the exposed active site on actin for a cross bridge. Figure 20.4 represents this. Stages in cross bridge formation, rotation of the head and breaking of the cross bridge. Here they have represented. First is sliding rotation. ADP combined with P and form ATP, breaking of cross bridge, myosin filament, actin filament, P and ADP. Here a cross bridge, myosin head, cross bridge formation. This pulls the attached actin filament toward the center of A band. The Z line attached to these actins are also pulled inward, thereby causing a shortening of the sarcomere. That is contraction. It is clear from the above steps that during shortening of the muscle, that is contraction, the I bands get reduced whereas the A bands retain the length. The myosin releasing the ADP and phosphate P goes back to its relaxed state. A new ATP binds and the cross bridge is broken. The ATP is again hydrolyzed by the myosin head and the cycle of cross bridge formation and breakage is repeated, causing further sliding. The process continues till the calcium ions are pumped back to the sarcoplasmic cisternae, resulting in the masking of actin filaments. This causes the return of cell lines back to their original position, that is relaxation. The reaction time of the fibers can be vary in different muscles. Repeated activation of the muscle can lead to the accumulation of lactic acid due to anaerobic breakdown of glycogen in them, causing fatigue. Muscle contain a red-colored oxygen-storing pigment called myoglobin. Myoglobin content is high in some of the muscles which give a reddish appearance. Such muscles are called the red fibers. These muscles also contain plenty of mitochondria which can utilize the large amount of oxygen stored in them for ATP production. These muscles therefore can also be called aerobic muscles. On the other hand, some of the muscles possess very less quantity of myoglobin and therefore appear pale or whitish. These are the white fibers. Number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is high. They depend on anaerobic process for energy. Let us see the skeletal system. Skeletal system consists of a framework of bones and few cartilage. This system has a significant role in movement shown by the body. Imagine chewing food without jaw bones and walking around without the limb bones. Bone and cartilage are specialized connective tissue. The former has a very hard matrix due to calcium salt in it, and the latter has slightly pliable matrix due to chondroitin salt. In human beings, this system is made up of 206 bones and a few cartilages. It is grouped into two principal divisions, the axial and the appendicular skeleton. Axial skeleton comprises 80 bones distributed along the main axis of the body. The skull, vertebral column, sternum, and the rib constitute axial skeleton. Skull 20.6 represent this is composed of two sets of bone, cranial and facial. The total of 222 bones. Cranial bones are 8 in number. They form the hard protective outer covering, cranium for the brain. The facial region is made up of 14 skeletal elements which form the frontal part of the skull. A single U-shaped bone called hyoid is present at the base of the buccal cavity, and it is also included in the skull. Each middle ear contains three tiny bones, malleus, incus, and stapes, collectively called ear ossicles. The skull region articulates with the superior region of the vertebral column with the help of two occipital condyles, dicondylic skull. Let us see the figure which is representing human skull. Here they have represented the frontal bone and parietal. Side two bones are temporal bones. Bottom bone is occipital, then occipital condyle, hyoid bone, mandibles, which is and maxilla, zygomatic bone, nasal bone, lacrimal bone, ethmoid bone, sphenoid bone. Let us see our vertebral column. 20.7 represent vertebral column. Right lateral view. Cervical vertebra, thoracic vertebra, lumbar vertebra, intervertebral disc, sacrum and coccyx. Our vertebral column is formed by 20 serially arranged units called vertebrae and is dorsally placed. It extends from the base of the skull and constitutes the main framework of the trunk. Each vertebra has a central hollow portion, neural canal, through which the spinal cord passes. First vertebra is the atlas and it articulates with the occipital condyle. The vertebral column is differentiated into cervical, thoracic, lumbar, sacral, and coccygeal. Cervical are 7, thoracic 12, lumbar 5, sacral 1, and coccygeal 1. You have to remember 7, 12, 5, 8, 8 in this way. Region starting from the skull. The number of cervical vertebrae are 7 in almost all mammals, including human beings. The vertebral column protects the spinal cord, supports the head, and serves as the point of attachment for the ribs and musculature of the back. Sternum is a flat bone on the ventral midline of thorax. There are 12 pairs of ribs. Each rib is a thin, flat bone connected dorsally to the vertebral column and ventrally to the sternum. It has two articulation surfaces on its dorsal end and is hence called bicephalic. First 7 pairs of ribs are called true ribs. Dorsally, they are attached to the thoracic vertebra and ventrally connected to the sternum with the help of hyaline cartilage. The 8th, 9th, and 10th pairs of ribs do not articulate directly with the sternum but join the 7th rib with the help of hyaline cartilage. These are called vertebrochondral false ribs. Last two pairs, 11th and 12th, of ribs are not connected ventrally and are called floating ribs. Thoracic vertebra, ribs, and sternum together form the ribcage. Figure 20.8 represents the structure of ribcage, ribs and ribcage. For 1 to 7 are true ribs, and 8, 9, 10, 3 are false ribs, and 11th and 12th are called as floating ribs. Here they are represented as sternum, ribs, vertebral column. The bones of the limb, along with their girdles, constitute the appendicular skeleton. Each limb is made up of 30 bones. The bones of the hand, forelimb, are humerus, radius, and ulna, carpals. Wrist bone 8 in number, metacarpals. Palm bones 5 in number, and phalanges 14 in number. Figure 20.9. Femur, thigh bone, the longest bone. Tibia and fibula, tarsal, ankle bone 7 in number, metatarsal 5 in number, and phalanges, digit 14 in number, are the bones of the legs, hind limb. Figure 20.10 represents this. A cup-shaped bone called a patella covers the knee ventrally, kneecap. First here they represented the right pectoral girdle and upper arm, clavicle, scapula, humerus, hand part, radius, ulna, carpal, metacarpal, and phalanges. Second figure represents the right pelvic girdle and lower limb bones, frontal view, ilium, pubis, ischium, coxal bone, sacrum, femur, patella, tibia, fibula, tarsal, metatarsals, and phalanges. Pectoral and pelvic girdles bones helps in the articulation of the upper and lower limbs, respectively, with the axial skeleton. Each girdle is formed of two halves. Each half of the pectoral girdle consists of a clavicle and a scapula. Scapula is a large triangular flat bone situated in the dorsal part of the thorax between the second and the seventh ribs. The dorsal flat triangular body of scapula has a slightly elevated ridges called the spine, which project as a flat, expanded process called the acromion. The clavicle articulates with this. Below the acromion is a depression called the glenoid cavity, which articulates with the head of the humerus to form the shoulder joint. Each clavicle is a long, slender bone with two curvatures. This bone is commonly called the collar bones. The pelvic girdle consists of two coxal bones, figure 20.10. Each coxal bone is formed by the fusion of three bones, ilium, ischium, and pubis. At the point of fusion of the above bones is a cavity called acetabulum, to which the thigh bone articulates. The two halves of the pelvic girdle meet ventrally to form the pubic symphysis, containing fibrous cartilage. Let us see the joints. Joints are essential for all type of movement involving the bony parts of the body. Locomotive movements are no exception to this. Joints are the points of contact between bones or between bones and cartilage. Force generated by the muscle is used to carry out movement through joints, where the joint acts as a fulcrum. The movability at this joint vary depending on different factors. Joints have been classified into three major structural forms, namely fibrous, cartilaginous, and synovial. Fibrous joints do not allow any movement. This type of joint is shown by the flat skull bones, which fuse end to end with the help of dense fibrous connective tissue in the form of suture to form the cranium. In cartilaginous joint, the bones involved are joined together with the help of cartilage. The joint between the adjacent vertebra in the vertebral column is of this pattern and it permits limited movements. Synovial joints are characterized by the presence of a fluid-filled synovial cavity between the articulating surfaces of the two bones. Such an arrangement allows considerable movement. These joints help in locomotion and many other movements. Ball and socket joint between humerus and pectoral girdle. Hinge joint, knee joint. You have to remember this joint. An example. Pivot joint between atlas and axis. Gliding joint between the carpals and saddle joint between carpal and metacarpals of thumb are some examples. Let us see the disorders of muscular and skeletal system. Myasthenia gravis, autoimmune disorder affecting neuromuscular junction leading to fatigue, weakening and paralysis of skeletal muscles. Second disorder is muscular dystrophy, progressive degradation of skeletal muscles mostly due to genetic disorder. Tetanic, rapid spasms, wild contraction in muscle due to low calcium in the body fluid. Arthritis, inflammation of joint. Osteoporosis, age-related disorder characterized by decreased bone mass and increased chances of fractures. Decreased level of estrogen is a common cause for osteoporosis. Gout, inflammation of joints due to accumulation of uric acid crystals. Let us see the last part of this chapter summary. Movement is an essential feature of all living beings. Protoplasmic streaming, ciliary movement, movement of fins, limbs, wings, etc. are some of the forms exhibited by animals. A voluntary movement which causes the animal to change its place is called a locomotion. Animals move generally in search of food, shelter, mate, breeding ground, better climate, or to protect themselves. The cells of the human body exhibit amoeboid, ciliary, and muscular movement. Locomotion and many other movements require coordinate muscular activities. Three types of muscles are present in our body. Skeletal muscles are attached to skeletal element. They appear striated and are voluntary in nature. Visceral muscles present in the inner wall of visceral organs are non-striated and involuntary. Cardiac muscles are the muscles of the heart. They are striated, branched, and involuntary. Muscle possess excitability, contractility, extensibility, and elasticity. Muscle fibers is the anatomical unit of muscles. You have to remember this point. This is not mentioned in the chapter but mentioned in the summary. That is, muscle fiber is the anatomical unit of muscle. Each muscle fiber has many parallelly arranged myofibrils. Each myofibril contains many serially arranged units called sarcomere, which are the functional units. Each sarcomere has a central A band made up of thick myosin filament and two halves I bands made up of thin actin filament on either side of it, marked by Z lines. Actin and myosin are polymerized protein with contractility. The active site for myosin on resting actin filament are masked by a protein, troponin. Myosin head contains ATPase and has ATP binding site and active site for actin. A motor neuron carries signal to the muscle fibers which generate an action potential in it. This causes the release of calcium from sarcoplasmic reticulum. Calcium activate actin which bind to the myosin head to form a cross bridge. These cross bridges pull the actin filament, causing them to slide over the myosin filament and thereby causing contraction. Calcium ions are then returned to the sarcoplasmic reticulum, which activate the actin. Cross bridges are broken and muscles relax. Repeated stimulation of muscles lead to fatigue. Muscles are classified as red and white fibers based primarily on the amount of red-colored myoglobin pigment in them. Bones and cartilage constitute our skeletal system. The skeletal system is divisible into axial and appendiculars. Skull, vertebral column, ribs, and sternum constitute the axial skeleton. Limb bones and girdles form the appendicular skeleton. Three types of joints are formed between the bones or between bones and cartilage: fibrous, cartilaginous, and synovial. Synovial joints allow considerable movement and thereby play a significant role in locomotion. In this way, we completed the reading of the chapter. If you like the video and to get more updates about this, so please don't forget to subscribe the channel Medical Deal. Thank you.